Welcome back guys. Now in the previous video we have seen how the insulin is released. Now in this video let's discuss what exactly this insulin doing. Okay, previously we have discussed how insulin is released. Okay, now insulin is released. Okay, done. Now what this insulin is doing? We all know insulin is supposed to decrease the blood levels of glucose. Okay, we have taken the food, now the blood levels of glucose is more. Now what we need to do? We need to transfer this glucose in the blood into the tissues. We have to move the glucose from the blood into the tissues. Okay, and that is mediated by insulin. How insulin mediates the transfer of glucose from the blood into the tissues? How? How means, see, now we have this insulin molecule, see, this is the insulin molecule. Okay, now insulin molecule is going to act on our tissues. Okay, mainly skeletal muscles. Okay, our skeletal muscles and the adipocytes. Insulin acts on skeletal muscles and adipocytes. So insulin is acting on its receptor. First of all, insulin is a peptide hormone. Okay, it's not a steroid hormone, so it cannot cross the cell membrane. So insulin receptors are present on the cell membrane, and these insulin receptors are the tyrosine kinase receptors. So insulin, see, this red molecule is the insulin molecule. Now this insulin molecule, it is coming and binding with its receptor, that is the insulin receptor, which is nothing but the tyrosine kinase receptor. Now once the insulin molecule is binding to its receptor, now what happens? See, there will be a lot of cascade, like there will be a cascade of reactions which are happening in the cell, but at the end of the day, what happens? At the end of the day, now see, this cell is a skeletal muscle, or you can take it as a adipocyte. Okay. Now when insulin is acting on this adipocyte or skeletal cell, now what happens? You see, in these cells, okay, in this skeletal muscle, already there is this vesicle. Okay, there is this one vesicle present. Now in these vesicles, what are these? There are these channels present, or there are these transporters present. So what is the name of this transporter? These transporters are called as a glucose transporters type 4, GLUT4. Okay, already these vesicles are preformed. These vesicles are containing these molecules. Okay, GLUT4 transporter molecules are present now. Now when you stimulate the cells with insulin molecule, now see, this is a transporter. Okay, now these transporters, okay, with the vesicle which are present in this vesicle. Now what is happening? See, now they are going to move towards the cell membrane. Now for the first time they are moving towards the cell membrane. See, previously there are no GLUT4 transporters on the cell surface. Okay, again I am repeating the point. Previously there are no glucose 4 transporters on the cell surface. Now when insulin is binding to its receptor, now this vesicle which contains the GLUT4 transporters, now they are moving towards the cell membrane. Now they are going to fuse with the cell membrane. So at the end of the day, now please concentrate. Now these transporters, now they are incorporated, and now they are planted into the cell membrane. Okay, so again, because of the action of insulin on the cell, at the end of the day, now you have glucose transporter type 4. Okay, these GLUT transporters are incorporated into the cell membrane. Now these are the glucose transporters. In our blood, who is there? In our blood there is a lot of glucose present. Lots and lots of glucose is present. Okay. Now this glucose, it's a big molecule. It cannot enter the cell. It cannot cross the cell membrane. Okay. Glucose cannot cross the cell membrane. Okay. Yes, glucose is more outside the cell. True. But still it cannot enter into the cell. Why? Because glucose is such a big molecule. It cannot cross the cell membrane. So for the entry of glucose, you need to have special proteins or special carrier molecules. These are the glucose transporters. Now, under the presence of glucose transporters type 4, now see these glucose molecules, now they are entering into the cell. Now they start to enter into the cell. See, now these glucose molecules are entering into the cell. So, glucose will be entering into the cell only under the influence of insulin. So, this is how, okay, this is how insulin will move the glucose from the blood into the tissues. So, insulin acts on the tissues, acts on the cells, makes the glucose transporters type 4 to move towards the cell membrane and incorporates the glucose transporters to planted in the cell membrane, so that glucose molecules are going to enter into the cell. All these glucose molecules are entering into the cell. Okay, with this we are done. Like how insulin is going to act on the cells. Now after this, let's discuss some important drugs, okay, which are used in the treatment of diabetes mellitus, especially type 2 diabetes mellitus. Okay, some pharmacological correlation. See, there are drugs called as sulfonylureas, okay, glyburide, glipizide, glyclazide, these are the drugs. Now these sulfonylureas, okay, they are considered as a insulinosecretagogues. If you take the sulfonylurea, more insulin will be produced in the body, more insulin will be secreted in the body. Okay, already insulin is there, but more insulin is secreted. So sulfonylureas, what they are doing? Why we are using these drugs in the treatment of diabetes mellitus is that the sulfonylureas, they will block the ATP sensitive potassium channels. Once the ATP sensitive potassium channels are blocked, means potassium is going to accumulate inside the cell, beta cell. Now this potassium will cause a depolarization. The depolarization will lead to influx of the calcium ions into the beta cells of the pancreas, and that calcium will move the vesicles which contain the insulin. Okay, that calcium which have entered into the beta cells, it moves the vesicles which contains the insulin, so that insulin containing vesicles are undergoing exocytosis. So exocytosis happens, insulin is released. So more and more insulin is released. Already we have discussed about this. Okay, already we have discussed this thing when we are discussing about the topic of how insulin is released. So sulfonylureas, see, they block the ATP sensitive potassium channels. When they block the ATP sensitive potassium channels, yes, more and more insulin is released. So sulfonylurea is considered as a secretagogue, insulin secretagogue. Okay, there are two important group of drugs which are insulinosecretagogues. One is sulfonylureas and meglitinides. Okay, I used to remember like SUMI, sulfonylureas, SU, sulfonylureas and ME, meglitinides. Okay, so meglitinides and sulfonylureas are the insulinosecretagogues. What they will do? They will block the ATP sensitive potassium channels, leading to more release of the insulin. Okay, done. Now after this, let's talk about a drug called as a diazoxide. Okay, now what is this drug diazoxide? What is the mechanism of action? Which conditions we are going to use this drug diazoxide? See, diazoxide is a potassium channel opener. Okay, it's a potassium channel opener. See, what is the relationship between potassium and insulin? Whenever the potassium levels increases inside the cell, what happens? Cell will undergo depolarization, beta cell will undergo depolarization, leading to the increased release of the insulin. Okay. Now, what is this diazoxide doing? Diazoxide, please concentrate guys. Diazoxide is a potassium channel opener. It opens the ATP sensitive potassium channel, so more and more potassium is going out of the cell. When more potassium goes out of the cell will be undergoing hyperpolarization. Potassium is going out, positive charges are going out, so cell will undergo inactivation or hyperpolarization. So, more insulin will be released or less insulin will be released? Less insulin will be released. Okay, so less insulin will be released. So, used in, say, decreases the insulin release. Okay, well and good. Now, we are going to use this diazoxide in which condition? So, where we are going to use this drug called as diazoxide? Diazoxide is used in the treatment of insulinomas. Okay, so diazoxide used in the treatment of insulinomas. So, what is this? Insulinoma, it's a tumor of beta cells of pancreas. Okay, it's a tumor. Insulinoma is a tumor of beta cells of pancreas which is releasing excessive insulin, excessive quantities of insulin is getting released. And whenever there is excessive insulin, that will cause hypoglycemia, and that can even cause a coma in the patient. Okay, because of the severe hypoglycemia, the patient can even go into the coma. So, in this person who is having insulinoma, what you have to do as a doctor? You have to decrease the insulin release. How you can decrease the insulin release? By giving drugs like diazoxide. So, diazoxide is used in the treatment of insulinoma. Why? Because diazoxide decreases the insulin release. Okay, well and good. Now, let's talk about the incretins. Incretin molecules like GLP-1, okay, glucagon-like peptide 1 or GIP-1, okay, gastric inhibitory peptide or gastric insulinotropic peptide, something like that. So, GLP-1 or GIP-1, okay, GLP, okay, GLP, glucagon-like peptide or gastric inhibitory peptide or gastric insulinotropic peptide, all these molecules are called as incretins, okay, incretins. Normally these molecules, see, whenever you take the food, okay, the food is getting digested inside your stomach, okay. Now, this glucose, okay, in the stomach, it will stimulate the release of this molecule. See, these incretins, yes, they are produced from the GIT, too, they are produced from the GIT. Now, what these incretins are doing? Now, these incretin molecules, okay, they will go to the pancreas, they will stimulate the pancreas to release more insulin, okay. These incretin molecules, say, they will increase the insulin release. They will increase the insulin release. Okay, they will increase the insulin release. So, my point here is how we can use these drugs. So, incretins, we know, incretins, they will increase the insulin release. So, in the pharmaceutical industry, okay, let's make drugs which are similar to incretins. So, incretin analogs. So, I have made a drug which is called as the incretin analog, exenatide, liraglutide. Now, these drugs, what they are doing? Now, these drugs when I take, they will also stimulate the pancreas to release more insulin. If more insulin is there, means that will be good in the treatment of diabetes mellitus. Okay, if more insulin is getting released, yes, we can use this property for the treatment of diabetes mellitus. We want more insulin. So, incretins, yeah, they are produced from the GIT. What they are doing? Increase the insulin release. Okay, well and good. But the problem is, see, these incretin molecules, GLP-1, GIP-1, see, they are acting on the pancreas. But their action is decreased Because this GLP and GIP, these molecules, the GLP and GIP molecules, they are broken down. Okay, very rapidly these molecules are broken down with the help of an enzyme called as dipeptidyl peptidase 4. See this GLP-1 or this GIP, they are rapidly degraded by an enzyme called as dipeptidyl peptidase. Okay, this happens physiologically. So what happens? The action of, okay, the half-life of the incretin molecules is decreased. This is not something good for the treatment of diabetes mellitus. See, for the treatment of diabetes mellitus, okay, we have to do two things. Especially with the incretin mechanism. We have to manufacture a drug. Okay, first we have to make a drug which is acting like incretin. Okay, so that is incretin analogs. We have to make drugs which are incretin analogs. So these incretin analogs, they will go and they will stimulate the pancreas to release more and more insulin. With the other hand, what we have to do is, we have to make a drug which will go and inhibit this dipeptidyl peptidase 4, DPP4. So if you inhibit dipeptidyl peptidase 4, what happens? Now the endogenous incretins, they will work for longer time. If they are working for longer time, more insulin will be released. Okay, so by using this concept, see, we have made two drugs. So that is incretin analogs like exenatide and liraglutide. So this exenatide and liraglutide are the drugs. Yes, they are used in the treatment of diabetes mellitus. Okay, they just act like incretin. They will go to the pancreas, stimulate the pancreas to release insulin. One of the important side effects of this exenatide and liraglutide is, see, these drugs will cause pancreatitis. Okay, so the side effect of this exenatide liraglutide, see, they are stimulating the pancreas, right? They might cause pancreatitis. And drugs like DPP4 inhibitors, okay, dipeptidyl peptidase 4, you have to inhibit that enzyme so that there will be more acting of the incretin molecules, more half-life will be there for the incretin molecules. So DPP4 inhibitors like sitagliptin, saxagliptin, linagliptin, vildagliptin, all these gliptins, these are the DPP4 inhibitors which are also used in the treatment of diabetes mellitus. But the side effect of these drugs is nasopharyngitis. Nasopharyngitis. So these are some important pharmacological correlations which I want you to know regarding the topic of insulin release, and how these drugs, okay, are going to be helpful in the treatment of diabetes mellitus. Okay. Now after this, let's discuss about the other actions of insulin. Okay, other actions of insulin. Now we all know that the primary action of the insulin is that insulin will act on its receptor in skeletal muscles and adipocytes, so that the glucose molecules will be going from the plasma, from the blood into the tissues, so that now tissues can utilize the glucose with the help of glucose transporter type 4. That's the major action. Okay, we are done with the major action. Not only that, see, this insulin will also stimulate certain biochemical processes and inhibit certain biochemical processes. How to understand this? See, the primary goal of insulin is what? The primary goal of insulin is to decrease the blood glucose levels. This is the primary goal. Okay, so all those biochemical reactions, which will decrease the blood glucose levels will be activated. Okay, there are certain biochemical reactions which will decrease the blood glucose levels. So all those biochemical reactions are activated, and those biochemical reactions which increase the blood glucose levels, they will be inhibited. Okay, so now first look at here guys, see, insulin, see, it is stimulating the glycogenesis. What is glycogenesis? Glycogenesis is happening in the liver. What is this glycogenesis? Conversion of the glucose into glycogen. Okay, conversion of the glucose into glycogen. So insulin, what it is doing? It is stimulating the glycogenesis, so that from the blood glucose molecules are taken into the liver. The glucose will be converted into glycogen and stored in the liver. So what happened to the blood levels of glucose? Decreases. Okay, blood levels of glucose is decreased. Okay, achieved, target achieved. Not only that, lipogenesis. Okay, or lipogenesis. What is lipogenesis? If you have enough amount of glucose, okay, that glucose will be converted into fat and stored in the adipocytes. So taking the blood glucose, converting it to fats and storing in the adipocytes. So blood levels of glucose is decreased. And glycolysis. What is glycolysis? Insulin is stimulating the glycolysis. So insulin will act on the skeletal muscles. Now what the skeletal muscles are going to do? Skeletal muscles will immediately take the glucose into them. Now this glucose will be going into glycolysis, oxidative decarboxylation of pyruvic acid, Krebs cycle, electron transport chain, ATP is generated. So at the end of the day, glycolysis means taking the glucose from the blood into the cells. Now in the cell, this glucose will be converted into pyruvates and at the end of the day, it will be used for the production of the ATP. Okay. So, all those reactions which decreases the blood glucose levels, they are activated. And those reactions which increase the blood glucose levels, for example, say glycogenolysis. If you cause the glycogenolysis, what happens? Glycogen will be broken down into glucose, glucose will come into the blood levels of glucose will be elevated. So, glycogenolysis is inhibited. Okay, and gluconeogenesis means whenever you are doing a fasting, for example, for 18 to 24 hours, okay, when you do fasting, what happens? The non-carbohydrate sources like proteins and fatty acids, like they will be used in the generation of the glucose. So, generation of glucose from non-carbohydrate sources, that is the gluconeogenesis. So, at the end of the day, the proteins or the amino acids, I should say, the amino acids and fatty acids, they will be utilized for the production of glucose. So, it will cause this process of gluconeogenesis will elevate the blood glucose level. It will try to elevate the blood glucose levels. So, that will be inhibited. So, insulin inhibits the gluconeogenesis. And not only that, say insulin inhibits the lipolysis. Insulin is an anabolic hormone. It stimulates the lipogenesis. So, see here, lipogenesis is stimulated so that glucose will be converted into fats. Okay, so insulin inhibits the lipolysis. Insulin is not a catabolic hormone, it's an anabolic hormone. So, stimulation of lipogenesis but inhibition of lipolysis. And very important point is, just for now remember this one point, later we will discuss in detail about what is this diabetic ketoacidosis. Okay, we will discuss about that later. But for now, say insulin, it inhibits the ketogenesis. Ketone bodies, they are formed inside the liver. The ketone bodies are high energy molecules. Okay, even ketone bodies will help in the generation of ATP. Okay, see insulin, what it will do? It will inhibit the ketogenesis. Okay, it will inhibit the ketogenesis. Okay, in the liver. So, till the time insulin is there, ketogenesis is not possible. Ketogenesis is not happening. Okay, so insulin inhibits the ketogenesis. These are some important actions. These are some important actions of biochemical actions. Like insulin activates certain biochemical processes, inhibit this other biochemical process. Not only that, when it comes to the ions, insulin, it will move the potassium ions into the cell. Okay, when you take the insulin shot, potassium will move into the cell. Okay, potassium will go into the cell. This is one of the important effect of the insulin. So, this property is used in the treatment of hyperkalemias. Okay, so hyperkalemia means what? More potassium in the blood. Okay, when you have more potassium in the blood, that can trigger the arrhythmias. Okay, that can trigger the arrhythmias that we don't want now. Okay, we don't want that arrhythmias now. So, if you want to decrease the blood levels of potassium, what you can use? You can use drugs like insulin. Why? Because when you take the insulin, all the potassium will be going into the cells. They will be going into the adipocytes especially. Okay, so all the potassium is now packed inside the cell. So, the blood levels of potassium will come down. So, these are some important actions of the insulin. Okay guys, so with this, we have seen what are the important actions of the insulin. Now, in the next video, we will discuss much about the insulin. Hope the video is helpful. Thank you.